and welcome everyone. So my name is Kai Kupferschmidt. I'm a journalist at the journal Science, and it's a pleasure to be here. I studied molecular biomedicine, and when you do that, you learn all of these amazing facts, right, about cells and our bodies and about nature. And one of the things that I find mind-blowing about this particular conference, this meeting, is that you end up meeting, a lot of the people who kind of were the first to find out these facts. I mean, these people basically created the knowledge that you tried to learn the night before an exam. And this is not a lie. I actually remember the lecture where I first saw a picture of an enzyme called ATP synthase. This is just an amazing piece of molecular machinery that basically is the molecular motor that drives our bodies. It's the reason that all of us managed to get up out of bed this morning, at least all of us who are here. And it's just a really remarkable piece of molecular machinery, and so it's a huge pleasure to be able to introduce Sir John Walker to you, who is actually the first person to see this beauty and was the person to explain the mechanism of this molecular motor. He won the Nobel Prize for Chemistry for this work in 1997, and we will return to ATP synthase in a little bit, but the topic of our talk today is one of the big challenges facing humanity. It's microbial resistance. Over to you.
We got another big challenge. So, as you heard in the introduction, what I'm interested in and have been for the last 35 years is biological energy conversion, particularly how do mitochondria work. But along the way, I've realized that there were opportunities to apply that rather specialized academic knowledge in a practical way. And so I've talked before about this. There are opportunities for us with our knowledge to contribute to the energy crisis. We're also interested in neurodegenerative diseases. And today what I want to talk about is the interface with antimicrobial resistance. And I may say these are global problems facing your generation, and they're challenges for you to take up, the next generation of young scientists. So let's go back to 1928. This is a reprise of Alexander Fleming's famous experiment where this mold adventitiously landed on this agar plate and prevented the growth of Staphylococcus aureus. So this was a demonstration of the effect of penicillin against a clinically important microorganism. But strangely enough, there was a lapse of 11 years before anything happened. So Fleming actually did nothing beyond report his original observation. And in 1939, Howard Florey and Ernst Boris Chain at Oxford University decided to start studying penicillin to try and see if it would be useful for clinical medicine. And they had to start at the beginning and produce the penicillin, isolate it. And the man who later became my PhD supervisor, Edward Abraham, contributed hugely to the structure of penicillin. But the important point here was that just 1 year after they began to study penicillin, Abraham noticed that there were Staphylococcus aureus that were resistant to penicillin. So the penicillin-resistant Staphylococcus aureus were already there. And of course, what the antibiotic is doing is to select, to kill off the sensitive ones and leave the resistant ones. And remarkably rapidly, given that this was wartime, penicillin was used clinically in battlefields in North Africa in 1943 and 1944. Now, if you look at the second line there, you'll see that in clinical practice, resistance was observed in 1946. And if you were to go down that list of the major antibiotics that have been introduced into clinical medicine, the general trend that you see is that quite soon after introduction into clinical practice, then resistance is observed. And these are examples of relentless rise of antibiotic resistance to Staphylococcus aureus, the blue line, vancomycin-resistant Enterococcus, the green line, and fluoroquinoline-resistant Pseudomonas. And now the chief medical officer for health in Britain has become concerned about this particular issue and has spoken extensively about it, and so has the World Health Organization. And so they put out this statistic that shows that at the current time, antimicrobial resistance, AMR, is somewhere behind road traffic accidents in terms of lethality. But they predict that by 2050, 10 million people per year will die from antimicrobial resistance unless we do something about it, and that's far more than the people that are dying currently from cancer. And those deaths will not be spread evenly over the world. The brunt will be borne by Asia and Africa, and the rest of the world will still be affected significantly. And the WHO also discussed how antibiotic resistance spreads. This says overprescription of antibiotics. Actually, it's inappropriate prescription of antibiotics, where doctors give antibiotics for viral infections where they're inappropriate. The second one I don't understand, and I'm going to ignore that. The third one on the top is that it's certainly one of the problems is the overuse of antibiotics in farming. And that's a very emotive issue, and farmers get very upset if you suggest that they should reduce the amount of antibiotics that they're using prophylactically. But undoubtedly it does contribute to the selection and propagation of resistant strains of bacteria. And then, of course, there are practical problems in hospitals, poor infection control in hospitals and clinics, lack of hygiene. And another related question that I'll come to in a moment is there's a lack of new antibiotics being developed. So this just gives you some impression of the amount of medically important antibiotics that are actually used in farming. 70% in animals, 30% in humans. And the actual amounts are something like 6 million kilograms, 7 million kilograms in animals and 3 million in humans. Now, this is what I was referring to. So in the 40s, 50s, and 60s, there were many antibiotics were discovered and brought into clinical medicine and had a huge impact. The cephalosporins, which again were discovered by my PhD supervisor, Edward Abraham, were introduced into clinical medicine in the 1960s. And after that, very little happened until 2000. And that allowed, again, resistance to build up against all the main armament in the clinical cabinet. And it's only in recent times that this problem has been taken up again at all seriously. And today, these are the 10 most dangerous antibiotic resistance bacteria. And many of them are familiar to you. Neisseria gonorrhea, Staphylococcus aureus, sepsis. And the one I want to focus on is Mycobacterium and tuberculosis. So tuberculosis is a disease of the respiratory system. It can spread to other tissues, the brain, kidneys, and bones. It gets spread by coughing. The symptoms are pain, fatigue, weight loss, fever. 2 billion people in the world are infected, and 2 million die annually. So this is a massive global problem, and 60% of those who are untreated die. And there's been a resurgence of TB, and it's partly poor adherence to TB control programs. The clinical measures that are taken require a long-term adherence to drugs, even up to 6 months. And this can be very difficult for people to pursue. And then again, there is this emergence of drug resistance, multi-drug resistance, extreme drug resistance, and now total drug resistance. In the 10 cases have now been identified in Asia with totally resistant to all known drugs. And there's a synergy with AIDS, with HIV. Immune-compromised people wake up a latent form of the disease that I'll just describe to you in a moment. And there's no quick way of diagnosing the disease, and drugs and vaccines are outdated. So this is what I was talking about. So the mycobacteria can go into a cryptic latent state where they form what's called a granuloma, and that's depicted there on the under dormancy. And you can see that the granuloma is surrounded by T cells and B cells and encapsulated in the middle, and they just sit there and they don't do anything, but they are alive. And what happens is that the granulomas accumulate lipids, and it's like a creamy cheese inside. But under particular conditions, and one of those conditions is becoming immune compromised, the mycobacteria wake up. So the people who got dormant mycobacteria are perfectly healthy. They can pursue a normal life, and many antibiotics will simply kill off the ones that are replicating and alive, and they don't touch the mycobacteria that are hidden in these granulomas, and so they can wake up, and then the disease starts again. So how can we control TB? Well, we clearly need new drugs against novel targets, and also new vaccines. So the vaccine that's most widely used is BCG, and that was introduced by the Pasteur Institute in 1921, and again, this topic of diagnosis. Now, there's one exciting breakthrough in this rather daunting field, and this was announced 10 years ago, a diaryl quinoline drug active on the ATP synthase of Mycobacterium tuberculosis. So this awakened my interest, of course. And the drug is called bedaquiline, and it's the first anti-TB drug with novel action to be introduced into clinical medicine in 40 years. And what it does is to inhibit the ATP synthase by preventing the rotation, the turning of the rotor. It's very specific for mycobacteria, and importantly, it's active against both the replicating and dormant states, the Mycobacterium, and it's also active against multiple resistance, totally resistant, and extremely resistant mycobacteria. So it looks like a wonder drug, but actually it isn't. It was rushed into clinical medicine because of the dire need for it by the FDA, based on phase 2 trials involving only 440 people. There are side effects. It can cause death, unexplained cases. There's cardiovascular risk, and it's recommended only for these extreme cases of multi-extreme and totally resistant Mycobacterium. It's also a very lipophilic molecule and stays in the human body for up to 6 months. And what happens then is that the drug accumulates in lysosomes, and people develop what is called phospholipidosis, which is somewhat like Neumann-Pick disease. It's a lipid storage disease. So the side effects can be quite dire. So I mean, clearly, what has happened here is that the ATP synthase has been identified as a drug target where one can make selective medicines that affect the mycobacterial enzyme, but not the human one. And so the view that I've taken is that we should try and follow this path and find more inhibitors. Now, this is the human enzyme that I've worked on for a very long time. It's a dimeric structure with a rotary action, which you're familiar with. I won't go through all of that. And we now know a lot of so this is from cryo-EM. From recent experiments, we now know a lot about how rotation is generated from what we call proton motive force, which is effectively a voltage driving this motor. Now, the important thing is that although the bacterial ATPases and the human ones, bacteria on the left, human on the right, operate in a similar way, nonetheless, there clearly are significant differences. If you look on the right of these two structures, you can see there are different proteins. There are additional proteins associated with the lower part, which is the membrane domain of the enzyme. And then the brown part is actually the rotor that's turning round at about 200 per second. And in humans, that has got eight identical components, and in bacteria, anywhere from 10 to 15. And in fact, the Mycobacterium has 9. So we actually know, although we know a huge amount about the human enzyme, we know very little about bacterial ATP synthasers. And you have to remember that bacteria live in very diverse physiological conditions. They operate anywhere from, they can operate anywhere from pH 2 to 10. They may live in the absence or presence of oxygen. Some of them live in the presence of high salt. Some of them are driven by protons. Others are driven by sodium ions. Some of them can make and hydrolyze ATP. So, for example, the one in E. coli can operate in both directions. In anaerobic conditions, it will hydrolyze ATP made by glycolysis. In other organisms, can only synthesize. So there must be regulatory mechanisms, and they're ill-defined. We really don't understand how these enzymes, when we look at them by X-ray crystallography, actually we can't understand why this particular one can operate in both directions and this one can only operate in one. So there's something missing. And then I've mentioned the differences in rotor sizes. And there they are. So these are actually gears in the motor, and the C8 one is the human one. C9, not there, is Mycobacterium. So this drug, bedaquiline, actually binds to this rotor ring, and it's able to sense the difference between the curvature of the C8 human ring and the C9 Mycobacterium ring. So what we need to do is to determine the overall structure of the ATP synthase. We're working on it. We're trying to understand why it can only make ATP and not hydrolyze it. How is it inhibited? And work out this inhibitory mechanism. There's the latest picture of where we've got to. It's actually the toughest X-ray structure I've ever worked on. So I'm coming to the end. So I want a slightly philosophical life as circular element. So in 1994, we described the molecular basis of rotation in ATP synthases, and then on a more personal life level, in the 1960s, I actually studied peptide antibiotics at Oxford with E.P. Abraham. And here, from 2013, we've been trying to provide the basis for new antibiotics to combat TB in Cambridge. So I've always liked this poem by Robert Frost. We dance round in a ring and suppose, and the secret sits in the middle and knows. Thank you very much.
Thank you very much. So before I open for questions, just a couple of questions for me. And you talked about the fact that there was a gap in basically the production of new antibiotics. A few came afterwards. But of course, one of the problems we face with antibiotics is that the way that the drug market works, it's not very profitable for a company. If they come out with a new antibiotic tomorrow, it would probably be reserved for very resistant cases, so it wouldn't be used much. If it's used for a week or so. If you come out with a new drug that lowers blood pressure, you can give that to people for 40, 50 years in rich countries. So how do we start to even address that structural problem? Is there something that scientists can do about that? Because you can't take a drug to the market yourself.
I think what we can do is to identify potential drug targets. We can even get involved in design of inhibitors, which will then, of course, need to be taken to a further stage if they're to be developed into antibiotics. And there are enterprises associated with academic institutions that actually do this kind of screening now. Once you've got to that point, actually, the Gates Foundation are quite interested in providing funding. So they recognize that there is a problem here in actually bringing these fundamental discoveries into something that's practical and useful. And they are prepared to put vast amounts of money into that final stage of development.
There are some ideas like, having a tax on antibiotics that could be used to develop new antibiotics, or extending the patent protection for new antibiotics. Is there a space here where governments actually need to act in some way, or do you think people like organizations like the Gates Foundation can basically make up for that?
I don't think we can expect the Gates Foundation to solve the problem on their own, and political assistance and tolerance of the kind you suggest may well be helpful, but I can imagine that there might be opposition to extending patents in specific instances.
I mean, the other question I wanted to ask is, even if we do find new antibiotics, and it's clear, there are still some targets we have, but there is an argument to be made that there is a finite number of these, and that we basically have to look at antibiotics as a kind of natural resource that we're slowly using up. When we use them, we get resistance, even if we take out, the use in animals. We will get resistance in humans probably. So at some point, we may just face a natural barrier to having new antibiotics. So in the long term, do we also need to find other strategies? Are there other ways of addressing?
Well, I mean, first of all, we've not really explored vast areas of the earth for potential natural products. Most antibiotics actually originate from the soil, and it's only latterly that people began to look in other ecological niches, in the oceans, for example. So there probably still is a lot of opportunity for exploration of the kind that was done in the 50s and 60s, and certainly activities in that area have reawakened. But there are also other strategies that are being pursued. The use of phages, for example, has been pursued in what was the Soviet Union, now in Russia and Georgia.
So these would basically be viruses that
These are bacterial viruses, which would be specific for particular bacteria, and some success is reported there, but I'm not aware of any extensive trial of phages in Western Europe or North America.
I'd love to open it up. There's a lot of people here. I'm sure there's also a lot of questions. Yes, let's start right here at the front. If you wait a second, the mic will come.
Thank you for the opportunity to start. My name is Dominic from Kenya. I'm a postdoctoral student at the African Academy of Sciences. Besides that, I'm a veterinary surgeon. And one of the issues that we deal with in Kenya is the problem of drug residues in milk and in meat. Probably, I don't know, what are your thoughts? Is there a way as scientists we can develop kits to test somehow? Because this is to test these drug residues before we consume the meat, because that seems to be an area, a source of this antimicrobial resistance. Number two, I really don't want to get to the politics, but there's a rumor that it's not that we don't have new antimicrobials drugs. It's just that some companies have decided to hide them. Because if then they bring them out, they'll be out of business. Maybe you can mention something about that. And then lastly, I believe that we should go to the direction of natural products. I work with medicinal plants against cancer, and some of the plants that I realized that have very good activity against cancer, I just tried to test them against gram-negative and gram-positive bacteria, and they worked very well. So probably if we go that direction, just as you said, we might get new novel compounds that can be used against the resistant microbes. Thank you.
Well, let me start at the end. So don't get your hopes up too high with the antibiotics you find yourself. So I worked on an antibiotic for 4 years as a PhD student at Oxford. It was a brilliant antibiotic. It killed off Staphylococcus aureus. Very low levels of resistance emerged, but when it was introduced into humans, it was immediately inactivated in the liver. And that was the end of that one. And so whilst my supervisor, Edward Abraham, was accumulating millions and millions from his royalties on cephalosporins, mine went nowhere. So that's the first point. The second point, moving backwards, I really can't comment on the behavior of drug companies. They may or may not be holding things back. I really have no knowledge of that point. The first point I couldn't hear completely, but I think it may have related to me. That's because I have some problems with my hearing. It may have related to meat, and I think one of the things that we can do actually is to reduce the amount of meat we consume. If we halve the amount of meat we consume, that would mean that fewer cattle and other animals would need to be raised. This would lower the consumption of antibiotics, and this would contribute to solving this problem. And some countries, actually New Zealand, now project by 2030 they will have removed, by taking measures of that kind and others, they will have removed completely the antibiotic use from farming. So it is possible to do that, but there doesn't seem to be much move in that direction in Western Europe and North America that I'm aware of. I'm sorry, I hope I answered your questions. Thank you very much.
Actually, if I can comment on the second point, because it's something that obviously as a science journalist we get a lot from readers. I think it's slightly based on a misconception of how drug companies work, because on a very basic level, to know that you actually have a functioning antibiotic, you would have to go through three phases of clinical trials that cost millions and include thousands of people. Nobody would do that if all they were planning to do was then to put it back into the drawer, I think. So, and today, of course, we also have trial registries. So, you know, if there are trials going on with certain substances, we should know about it. I mean, there's lots of problems with that as well. So, yeah, so it just seems a very unlikely scenario, especially with antibiotics. You know, once you have it, you know, you could start using it immediately and make at least some money of it. The problem is that the incentive is so low because you make so little money with it that nobody's investing in it in the first place. They'd rather bring out the 10th drug to lower blood pressure, for instance. Sorry. Yes, over there. Sorry. Go 1, 2. Yeah. It's okay. A microphone is coming. Don't worry. Yeah. Thanks.
So, thank you very much. My name is Leopold Chenche. I'm a postdoctoral scientist at the London School of Hygiene and Tropical Medicine, based at the unit in the Gambia. My question relates to alternative treatment for tuberculosis. We have all heard the difficulty to generate antibiotics that target only the bacteria. We also know that tuberculosis really expands as an overreaction of the immune system to the bacteria, causing immunopathology. And still, the vaccine that we use are not very effective because we really don't understand the immunology very well. Is it not an area to explore deeper to understand the immune checkpoints that the bacteria use to be able to survive in this hostile environment, so that we can use possibly repurposed drugs, even from the cancer field, to start using as what we now call host-directed therapy. So help the immune system to participate in the clearing of the bacteria. Such treatment could be adjunctive treatment to the antibiotics and could contribute to reduce the length of treatment and increase compliance and reduce actually resistance that are generated during the course of treatment. Thank you.
Can you summarize that for me?
Yeah, sure. That's a great question. I mean, so the question was, is there a way of, you know, what if we don't actually attack the bacteria directly, but try to modulate the immune system, which causes a lot of the pathology in a disease like tuberculosis? So are there ways of dealing with tuberculosis that don't directly attack the bacterium?
Well, not that I'm aware of. Of course, I'm not a clinician. But I spoke about this major clinical problem with tuberculosis, and that is the encapsulation in granulomas and dealing with the latent state. So I think understanding the latent state in tuberculosis is very important. They're actually not totally cryptic. It's not like a spore. There is actually biochemical activity going on, but this is an area that's been very studied, little studied. There's actually a poster here on a related topic from people at Oxford University, which I need to look at. And so I think those are the areas to concentrate on.
I mean, there's also sometimes this idea is brought up that rather than develop completely new antibiotics, you can try to have an antibiotic where there's resistance and then try to find a substance that just kind of circumvents the resistance, right? So that together maybe like a resistance breaker. Is that something that's...
So, I mean, to take an example, resistance to penicillins is due to beta-lactamase penicillinase. So what you're suggesting is that you develop other inhibitors of penicillinase, which then would allow the antibiotic to do its job. I guess that's possible, but I don't have any particular example or any sustained effort to go down that route. And then probably the bugs would anyway become resistant again to the new compound. So I think a much more useful strategy actually for use of antibiotics is what has been done in the treatment of AIDS, and that is to use combination approaches and to use more than one compound at a time to attack a drug. And in that way you can retard the emergence of antibiotics to a particular compound.
Right.
So I think those strategies actually are more important.
Yes, right here in the front.
Thanks for a very interesting talk. I'm working in the field of the gut microbiome, especially in child malnutrition in sub-Saharan Africa. And there's good evidence that mass administration of antibiotics can reduce child mortality. And people like Gates are very happy to fund these big studies, but the World Health Organization are quite reluctant to adopt these approaches due to antibiotic resistance. So I'm wondering what your thoughts are on kind of these ethical considerations where antibiotics still can be very, very useful for example, preventing child mortality, but we still have this growing threat of antimicrobial resistance. And in the Western world, there's not really a big move to preventing the overuse of antibiotics for simple things such as simple infections, whereas they can really be useful for reducing child mortality.
Well, I mean, I think clearly one should do whatever one can to avoid and reduce infant mortality. But I think we do have a responsibility to try and reduce usage and exposure of microorganisms to these drugs so that their efficacy is sustained for a longer period of time. So I think that those strategies are extremely important.
Yes, over here.
Thanks a lot. Actually, I'm a clinician from Austria, from Vienna. And I would like to tell all the other clinicians, please educate your patients that they take the drugs as scheduled. Often, antibiotics are pre-terminated before the scheduled time, and then these resistance may arise. A simple diarrhea can be easily treated with, for instance, yogurt or something else. And I just wanted to say this factor which contributes to these resistances in our field. Thank you.
Thank you very much.
Questions? Yes, over here. Sorry, behind you. Come to you afterwards.
Sorry.
No, yeah, go ahead. With
Okay.
you, yeah.
There was actually a beautiful study by a group at EMBL, Tupas and Borg, who found that once they screened 1,000 drugs that are actually non-antibiotic, that a quarter of them did kill specific strains in the gut microbiome. Meaning, and I mean, you can spin this story now and say, okay, this actually has a very dramatic effect on microbial resistance in general, just by taking drugs for a really long time that each of us needs for different reasons. So how do you think, is that worth something to follow up on, to kind of identify how they have effects on bacteria in general? And is that something to consider also to tell your patients maybe to not go and take everything that's maybe not necessary in the first place?
Well, I'm not aware of these effects, but they sound extremely interesting. Sounds like something that should be followed up, and to find out what the fundamental basis of it is. So use your curiosity and try and find out.
Okay, yeah, we have one. The mic is coming. We'll
Very short. I
Okay.
can speak loud. The why is not the most straightforward strategy, given this very potent inhibitory and selective inhibition of the tuberculosis motor, why is not simple SAR on that compound focusing on the side effects, not the most straightforward strategy?
SAR.
Structure activity, in other words, modification of the chemical, synthesis and modification, and then focusing on retention of the inhibitory effects and diminution of the side effects.
That is going on. So the compound was discovered by Johnson & Johnson, by Janssen's, which is part of Johnson & Johnson. So of course there's a lot of SAR going on that particular compound, but there is already a quite extensive resistance emerging to the existing compounds.
So I think we have time for one last question. We're a little bit behind schedule. Yes, over there, young woman in the last row. Yeah.
Thank you for this great talk. My name is Elena from the University of Melbourne. I just have a quick question. So with today's strict regulations, a lot of the antibiotics we're actually using wouldn't be approved anymore. So I'm thinking of colistin with like nephrotoxicity or neurotoxicity. Do you think the future of treating antimicrobial resistance is combination therapy of all the antibiotics with different mode of actions rather than investing all the money in developing new antibiotics?
Well, I was advocating using combinatorial approaches of different compounds. The idea of using antibiotics that have side effects, I guess you may do that in dire circumstances, and that's exactly what's happening in tuberculosis. These side effects are well known. It's known that they can cause damage or even death, but doctors calculate that it's worth the risk of using the drugs to try and deal with the problem. So these are very difficult decisions, I think, that clinicians have to make, and different evaluations that need to be made.
Thank you very much. I hope it's become clear that if anybody's still looking for an important problem to tackle, antimicrobial resistances are a huge one. So thank you everyone for being here. Thank you very much for the talk. Have a good break.